Good afternoon. Welcome to the ESHG webinar. I'm Bill Newman, as the Vice President of the ESHG. We're just going to give people a few minutes just to join the call for this live event. It's a program that we've started earlier this year. We've had a number of webinars and they're available on the ESHG YouTube channel, and the next talk will be available there in the coming days. We have a program that we have a webinar each month, with a couple of exceptions when holidays get in the way, and they're always pretty much the last Wednesday of the month. So it's an absolute pleasure today to cover a different topic that we've not covered before, but a really important topic, and a topic that has a window into the world of genomics. And we've got a wonderful speaker, Elfride de Baere, who's a professor of ophthalmic and developmental genetics. She's a physician scientist and head of the laboratory at the Centre of Medical Genetics in Ghent in Belgium. And Elfride has been a very active member of our scientific program committee for the European Society of Human Genetics, has had a long-standing interest in inherited retinal disease, and more recently has really been applying multi-omics approaches to truly understand the biology of this complex heterogeneous group of disorders, for which now excitingly therapeutic options are starting to emerge. And so without further ado, I'd like to hand over to Elfride so she can give her presentation. We will open a question and answers session. So towards the end of Elfride's presentation, the tab at the bottom of your screen called Q&A will allow you to post your questions. So rather than doing that in the chat portal, if you please do that in the Q&A portal, and then I will moderate questions to Elfride at the end of this webinar. So Elfride, the floor is yours. Thank you so much.
Thank you so much, Bill, for this invitation and for the kind introduction. Today, I would like to talk about multi-omics in inherited eye diseases, and I will highlight missing heritability, causes of missing heritability, and also how tackling missing heritability can open prospects for therapy. This is what many people experience, many people that have a hereditary eye condition that can affect the central vision and that can also affect the peripheral vision. Often it ends in legal blindness. So this is to illustrate that this is a very devastating group of conditions. I don't have any financial relationships to disclose for what I will talk about today. First of all, I would like to highlight the team with which I have the privilege of working with. This is the Eye and Developmental Genetics team. This is my research team, and I also closely collaborate with the clinical team. What do? We solve diagnosis in inherited eye diseases with an emphasis on inherited retinal diseases. We have a particular interest in the non-coding genome that is important for gene regulation, and I have a particular interest also in functionally conserved and sequence-conserved sequences residing in the non-coding genome. And then by doing this, we aim to advance precision medicine for inherited eye diseases, inherited retinal diseases, from diagnostics to also to preventive medicine to therapy. And today I would like to focus. The group of inherited eye diseases is a very large group. Many inherited diseases have an eye involvement. Today, I would like to focus on the retinal diseases and then also on the diseases affecting the optic nerve. So here you can appreciate retinal cells, the outer segment of the retina, which is the light-sensitive layer of the eye. we see the neuroretina, and then we also, underneath the neuroretina, you have the retinal pigment epithelium. Then the ganglion cells, the retinal ganglion cells, they come together in the optic nerve that sends signals to the brain. And this is a group of conditions I will talk about. This is a very heterogeneous group of conditions from a clinical point of view, but also from a genetic point of view. We know that there's a very recent article that appeared in American Journal of Human Genetics. We know that today over 200 non-syndromic disease genes are implicated in this group of diseases, and over 300 genes that have been implicated in syndromic disease. So this is a paper that just appeared in American Journal of Human Genetics, and that has been led by Carlo Rivolta. The database is called Retigene. So my talk will be relatively clinically oriented, and I would like to highlight what we do for standard of care genetic testing of inherited eye diseases in Belgium. We start from a request from a physician, and in Belgium this is often an expert ophthalmologist or a clinical geneticist with expertise in ophthalmic genetics. We receive a request, and often this is a request for a single patient, and we have sufficient clinical information, and then we do the standard of care. We do mostly exome-based testing, and we are shifting now to genome-based testing. We rarely do targeted gene panels, and in some instances where there is a very clear clinical diagnosis, we sometimes do single gene sequencing. And here at the bottom left, you can appreciate the panels that we offer. We put an in silico panel on the exome data, and this is the testing, the panel-based testing that we offer, and this is based on exome data with a shift to genome data. What can we identify using this standard of care? We identify single nucleotide variants and also copy number variants on the exome data. When doing this standard of care, this is pretty good in terms of diagnostic yield. So this is, on average, there is a diagnostic yield of 60% in the large group of patients with inherited eye diseases, meaning that there's 40% of patients that remains without diagnosis. And in that case, we are confronted with missing heritability in this group of diseases. And I list here several causes, not all the causes, but several causes of missing heritability. And today I will highlight some of these possible causes of missing heritability. I will talk about variants in novel candidate genes, sometimes also in old candidate or in old known genes. I will talk about different groups of different types of non-coding variants and also about structural variants, and I will also present a case of mosaicism. So first of all, I would like to highlight the integrated multi-omics approach that we are using now to tackle missing heritability in inherited eye diseases. Then I will zoom into data. I will present a recent example of how coding variants in a known retinal gene appear to be an important cause of missing heritability in our local cohort. Then I will talk about non-coding variants, different situations, and then I will end with some single molecule approaches that we use to interpret structural variants in inherited eye diseases. So let me start with the integrated approach. And the integrated approach started with a project called BSolve-RD. And BSolve-RD includes patients that have been seen by Belgian clinicians and that underwent standard of care genetic testing. Most of these patients have inherited retinal disease, and they all underwent standard of care testing. So all of the patients underwent whole exome sequencing. And in this group of patients, this is a group that is now, it's over 180 patients. It's a relatively small cohort. And then in this group of patients, we do whole genome sequencing, short read whole genome sequencing, and we look into coding SNVs. Then in the next step, in the second step, we look at non-coding SNVs in genes implicated in inherited eye diseases. Then we look at structural variants, and then in the fourth step, we look at candidate genes. So first of all, when we have genome data, then this is followed by an integrated multi-omics approach, and the goal is to unravel missing heritability in these patients, because these patients are of the group of 40 patients without a molecular diagnosis. So the first step is short-read whole genome sequencing, and then, if possible, and I will explain in what cases we do, RNA-seq, we do short-read RNA-seq. We have done that in this cohort. Then if RNA-seq on clinically accessible tissues is not possible, then we do in vitro assays, and these can be in vitro splicing, minigene assays, or these can be luciferase assays. So this is an RNA-based approach. And then in some cases, we also do a gene-tailored approach. I will also give examples of that. And in some cases, we need to reprogram to induce pluripotent stem cells and then differentiate these iPSCs to, for instance, retinal pigment epithelium or retinal organoids to be able to do multi-omics, such as RNA-seq or other omics. Making this an integrated approach, and it's an integrated approach, but it's also tailored for specific genes involved, for specific family context, and I will highlight this. So let me start with a recent example of variants in a novel candidate gene that leads to a novel, in this case, a novel retinopathy. And I will illustrate how an old visual cycle gene that is very well studied, the RPE65 genes, led to a new discovery of a new retinopathy in our Belgian BSolve-RD cohort. So you have probably already heard of RPE65. This is encoding a retinal pigment epithelium specific 65 kilodalton protein. And the function of this protein is an isomerase hydrolase in the visual cycle. And this is taking place in the outer segments and also in the retinal pigment epithelium of our eye. And this is well known because it's also very well studied, because this is a target for therapy in patients with biallelic recessive inherited retinal disease, and this is mostly congenital or early onset, and this was the very first gene therapy that was ever approved by the FDA. And the name of this gene therapy is Luxturna. This gene is known by many different variants, including variants of unknown significance, and most of the cases are recessive. In 2011, there was a group from Ireland that reported for the first time an autosomal dominant form linked to a variant in this gene. And this was a missense variant that was only found in few families in the iris population. It was a founder variant, and the presumed mechanism was an effect on splicing. So far, the mechanism has not been completely elucidated, and there's at this moment no therapy for this dominant retinopathy. Then, when we started off the BSolve-RD project, we did have whole genome sequencing data from the cohort of patients that underwent exome sequencing. And Miriam Bowens, who is pictured here, she noticed that there was a monoallelic RPE65 variant in whole genome sequencing data of 3 patients with a particular phenotype. They all were referred for the indication macular dystrophy and also the MIDD mitochondrial that was very similar to this MIDD phenotype. And she was wondering if the RPE65 variant, the monoallelic variant, had to do something with it. You can see the variant at the bottom of the slides. Then we looked back at our cohort of patients with retinal disease, patients that underwent whole genome sequencing. This led to the identification of 3 patients. We looked at our whole exome sequencing cohort. This revealed another 28 patients with this particular variant and also with similar phenotype. We did then a targeted testing of unsolved IRD patients who were reported with also a similar phenotype, which is actually a maculopathy of the central part of the retina. This revealed another 14 patients, and then we also did segregation analysis in family members, another 30 patients, and this led to a cohort in Belgium of 75 patients. Then we talked to our collaborators, our international collaborators, And this led then to, this revealed then another 10 patients with this particular variant and with a similar phenotype. I want to emphasize that in the UK, for instance, there are no patients with this. So in the UK Biobank, there are no patients with this variant. So we found mutations in the replication cohort with new patients from the Netherlands, France, and Canada. It appears to be a mutation that was found in Flanders. And you can see here the different countries where we have other patients. What is also remarkable is that the phenotype is, it could be subclassified in two recognizable phenotypes, a rather mild phenotype and then a more severe phenotype, a pattern dystrophy phenotype that you can see on the right. And this is, it's a dominant disease. It is late onset disease. And also the penetrance is complete. If the ophthalmologist looks at the retina, then the ophthalmologist does see a phenotype, but the patients don't always have complaints. So especially the modeled milder form, there the patients don't have yet complaints. So we also are confronted with a large inter- and intrafamilial variability. It is a dominant inheritance pattern with complete penetrance if you consider what the ophthalmologist observes as penetrance. Then we also did haplotype analysis using short-read and long-read sequencing, and we could identify a common haplotype of over 400 kb, supporting the initial hypothesis that this is Belgian or it's even a Flemish founder variant that was also found in other populations. So this led us to start off an integrated approach to validate the new dominant RPE65 retinopathy. The phenotype is different from the phenotype that was reported in the Irish dominant retinopathy. And what is interesting is that the phenotype is very similar. Also, the progress of disease is similar to age-related macular degeneration, which is a common complex retinal disease. So the gene is not expressed in clinically accessible tissue. The first thing that Eline van Voren, a PhD student in my team, undertook was in vitro analysis. First of all, we looked at the conservation of the amino acid, and you see here that it's located in a highly conserved domain, as well as the Irish variant is also very well conserved. Then we looked at the effect on the dimer structure of RPE65 and also at the monomer crystal structure of RPE65. And we saw that by the introduction of the mutant allele, there was a predicted repulsive effect of another allele in the monomer. Then Eline did several in vitro assays, as said. So we looked at protein expression, and we also included the Irish variant. We saw that what we also observed for the Irish variant, that there was a reduced expression, a reduced protein expression, and this could also be confirmed by the isomerase activity, which was also reduced. Co-immunoprecipitation was also done. And then we also saw that CETSA assay, which looks at stability, showed changed an impaired stability of the mutated protein. So as the gene is not expressed in clinically accessible tissue, it has a very tightly regulated, very specific expression in the retina, more specifically in the RPE. We decided it would be good to establish stem cells from patients with this retinopathy. So we reprogrammed patients, cells from 3 patients with this retinopathy to iPSCs, and we differentiated them. This is work from Eline Heens, a PhD student. So you can see here impaired pigmentation in one of the clones, one of the cells. Then also we could observe decreased PML17 expression and also decreased phagocytic activity, which shows impairment of the function in the RPE. And then we also see that there is impaired secretion, which all indicates impaired secretion of PDEF and VEGF, which all shows decreased RPE function. Why is this important? Because we think that this novel retinopathy is a target for therapy. And by these observations, including also transmission electron microscopy observations showing abnormal mitochondrial morphology, by putting together these observations, this allows us to put forward hypotheses about potential therapies that are pathway-based, that are gene agnostic, and this is our aim, to establish a pathway-based therapy. What we also did on the material the differentiated material, is we did transcriptome analysis, RNA-seq, and this revealed an altered antioxidant function and an altered oxidative stress regulation. And also these findings help us to put forward hypotheses about potential therapies. To have good quality control, we are also generating isogenic controls, and then the other way around, Eileen will also establish knock-ins of the particular Belgian retinopathy, but also of the Irish maculopathy. And then it's interesting to mention that apart from the Belgian population, we are also aware of novel dominant retinopathies linked to RPE65 in other populations. This also emphasizes the importance of ancestry. So this is a very local, very population-specific mutation, and this is also now appearing in other populations. Why do we think that this is a very interesting finding? Because of the similarities with the complex disease AMD, we consider this rare disease as a crowbar to find therapies that are also relevant for complex diseases. So this is our hypothesis. So to sum up, the integrated approach so far is needed to validate the new dominant RPE65 retinopathy. We are establishing a preclinical iPSC RPE model as soon as we have identified potential small molecules that can be used for therapeutic purposes. We will use this preclinical model. Of course, we will also further unravel the underlying pathomechanism, and then this preclinical model is excellent to guide us toward potential treatments. This was the first part of my talk. Then I will move on to examples of non-coding variation that solves monoallelic, half-solved, and also biallelic cases with inherited eye diseases. And this work has also been done by Mirjam Bouwens, together with Matthias van Heetvelde, who is also a postdoc in my team, and who is doing the bioinformatics and the data analysis, together with Quentin Mailleux, who has done the in vitro experiments, such as minigene assays. So in the cohort that we studied, we found several non-coding variants. And for some of them, it was possible, for some patients, it was possible to do RNA-seq, combined or not with in vitro minigene assays, to interpret non-coding splicing variants. The first example is WGS revealing non-coding monoallelic mutation or variant in OP1, which is the major gene for the dominant form of optic atrophy. And you see here a large pedigree with many affected individuals in which a deep intronic OP1 variant was found, and this variant appeared to segregate with the disease. So we have done segregation analysis. This you see at the right bottom, you see the patient retina and the optic nerve that is affected. We have done predictions, and these Splice AI predictions, for instance, revealed a pseudo-exon inclusion, which is often seen as a consequence of the deep intronic mutation. We have done RNA-seq on whole blood, which is possible because OP1 is expressed in clinically accessible tissues, such as blood. And this indeed did confirm pseudo-exon inclusion. And you can see that some material is treated with CHX and other material is not treated. And you see about the same pseudo-exon inclusion. And this can be explained because this is an in-frame insertion, and there you can expect no change, no NMD. Then a second case is a case where we revealed a non-coding monoallelic OP1 variant, also a deep intronic one, in a patient with optic atrophy. We did then RNA-seq on blood, and this confirmed a splice effect. And this also showed, in contrast to what we expected, a nonsense-mediated decay. And the reason is that we could see a difference, we could observe a 2 base pair difference compared to the splice AI predictions and also the mini-gene results, showing that there can be differences can be observed between in silico and in vitro assays and what you see in vivo here, in this case on blood. And this shows here this RNA-seq result that this is an out-of-frame insertion of 80 base pairs instead of the predicted 90 base pairs. Another example is this time not of non-coding variant with a splice effect, but a non-coding variant with a putative effect, a putative regulatory effect. Here, this is a promoter variant in the RPGRIP1 gene, again, in a half-solved patient with a nonsense variant. You can see the location of the promoter variant at position -152, and this is predicted to coincide with an OTX binding site, OTX, which is an important transcription factor of the eye. So to do an assessment in silico, we make use of our RegRed database containing many epigenomic signatures, and this is very useful to have a first idea about the potential effect of such regulatory variants. So here we also, apart from the predictions and the predicted loss of the OTX2 binding site, we also did luciferase assays combined with OTX2 addition, and this showed indeed an effect on gene regulation. And interestingly, here we could do RNA-seq, this time not on blood, but on fibroblast, because as you see at the right, the expression of the gene is high enough to do RNA-seq. It was not high enough to do that in blood. And this confirmed to start with pseudo-exon inclusion, which was in agreement with the predictions. And then on top of that, we could also see, we could also confirm the single gene deletion at the RNA level. You see that this is less abundant in patients compared to control exon 10. So taken together, this multi-omics approach in clinically accessible tissues reveals genetic diagnosis in over 44 of our pre-screened IED cohorts, which is quite a lot. And another observation is that coding SNVs in known genes make up over 8%. Non-coding splice and regulatory SNVs in known IRD genes and IED genes make up around 20%. Structural variants in known IRD genes are only 3%, which is a bit an underestimation. And then variants in novel candidate genes, IRD genes, make up about 40.5%. So this is clearly contributing to the diagnostic yield in these patients with inherited eye diseases. And then for the last part of my talk, I would like to zoom in on the importance of multi-omics and more specifically single molecule multi-omics approaches to interpret structural variants in inherited eye diseases. As you all know, it can be quite hard to identify structural variants. Even in short-read whole genome sequencing data, it can be tricky to identify cryptic or complex structural variants such as inversions, such as translocations and insertions, tandem duplications. And in that case, we need to rely on single molecule technologies such as long-read sequencing or optical genome mapping, which are all single molecule technologies. And by using these technologies, these approaches can help us forward to interpret structural variants in inherited eye diseases, because despite the introduction of exome sequencing, short-read whole genome sequencing, shallow whole genome sequencing for molecular karyotyping, we are confronted with a diagnostic gap, and we aim to close them using single molecule approaches. The first example is use of single molecule approaches to interpret a structural variant in a family with optic atrophy. And we see here a pedigree where a mother has bilateral optic atrophy, and she has a son who is also affected with the same condition. The mother still has a child wish, and she was referred to the consultation. Our standard of care testing revealed a duplication of exon 10 and 11, a duplication of OP1. We did not identify any other pathogenic variants, and the question was if this finding was clinically actionable. We have discussed that within our clinical staff, and we decided that there was some homework to do, that we wanted to know if this was a tandem duplication or an inverted duplication, because there the mechanism would be different. So we conducted, so this is here the location of the duplication, and then we decided to combine short read and single molecule multi-omics. We decided to do optical genome mapping, although we see that the duplication is relatively small. We also decided to do long read sequencing for more accurate delineation of the structural variant and to know how it was, if that was a tandem duplication or a more complex inverted duplication. And then we also looked at the RNA level OP1 is expressed in clinically accessible tissue. So this revealed, here you can observe the duplication, and it's a small duplication. It's a bit on the edge of the resolution of optical genome mapping, but we could see that this looked like, like a regular duplication. And then using long read sequencing, this confirmed that this is indeed a tandem duplication. You can nicely see that at the right, and this was from the maternal allele. So we also did RNA sequencing, and this showed that there was no reduced expression, suggesting that there is no NMD. So taken together, this multi-omics workup led to the confirmation of our hypothesis that this duplication is a tandem duplication, an in-frame duplication with a tentative dominant negative effect. We could classify this duplication as a pathogenic duplication, and we could report back that this would be a result that is eligible for pre-implantation genetic testing, which was also the wish of the family. Then another example is an example that has been completely established by Dr. Stijn van de Sompelen, who is working in the diagnostic team. This is a family in which two twin sisters, they have a diagnosis of optic atrophy. And they, many years ago, they underwent genetic testing, and that consisted then of sequencing and multiplex ligation-dependent probe amplification. And we could see more or less the same results in both sisters, but what we saw was quite difficult to interpret MLPA results with ratios that are not all 0.5. So that suggested potential different alleles, but then we didn't hear from the family for many years. As there was a son with mild disease, we also tested the son, and we observed that there was a deletion present in the son. We saw that there was a result that is compatible with the deletion of exon 22, 23, which is different compared to the results observed in the mother. And this then prompted us to do further workup in this family. We did optical genome mapping to disentangle the different potential alleles in OP1. And we could indeed see the deletion in the son using optical genome mapping. So this was the heterozygous allele. Then when we looked at the mother, we indeed saw 3 deletion alleles. We saw allele 1 that is also present in the son. We saw an allele 2, and then we also saw a 3rd allele. And you can observe that we could nicely also observe the allele frequencies, the VAF of these alleles, showing different alleles in the mother. This was also confirmed. So if we put together the results of optical genome mapping and the results of the MLPA, then this aligned well. And then on top of that, we also wanted to do long read sequencing to delineate the alleles. And you can see that these alleles in the mother can also be found using long read sequencing, the most prevalent ones, at least. And then we did RNA-seq, revealing the low frequency deletion in this time, and then also the 2 more prevalent deletion alleles. We did also RNA-seq in the son, and this did confirm the presence of the deletion of exon 21, 23. The twin sister has the same mosaic alleles. So it is our hypothesis that this complex mosaic structural variant occurred very early during development. And this family has child wish. They would like to move on with pre-implantation genetic testing. And I also want to emphasize that such a result has an impact on the how to progress with pre-implantation genetic testing, because you have to take into account these different deletion alleles with the mother. So to conclude, I have highlighted the combination of short read and single molecule multi-omics. I have highlighted a framework that can help to close the diagnostic gap in inherited eye diseases. Doing this, we identify the coding variants, but we can also identify non-coding variants with an effect on splicing, an effect on regulation, and also that can also represent complex structural variants. We combined the whole genome sequencing layer with the transcriptomics layer, and then also with tailored functional assays. And then in the future, we will also combine this layer with epigenomics layers. I did not highlight this today, but this is also a layer to take into account to assess the effects of structural variants, and also to look at, to combine genome and methylation layers to assess the effect of several variants. Why is an early diagnosis important in inherited eye diseases? Especially for retinal diseases, an early diagnosis is really important because there is a prospect of gene therapy. Gene therapy that can be at the DNA level, that can be at the RNA level. Also, pathway gene agnostic based therapies are emerging today, or modifier therapies, but it is very important to know the genetic diagnosis. If the disease progresses further, then also other types of therapy, such as cell therapy, are possible, retinal prosthesis that are possible in an end stage of disease. But again, an early diagnosis is very important for therapeutic reasons, but also, of course, for reproductive reasons, as I also highlighted today. To conclude, I have shown today that multi-omics, different layers of omics combined with genomics, improve the diagnostic yield. In our case, we had a diagnostic yield of over 44% in a pre-screened cohort with inherited eye diseases. A multi-omics approach is not only crucial to identify pathogenic variants, but also for variant interpretation. And then non-coding variants, non-coding splice variants are the most prevalent, together with novel disease genes in our cohort, are important contributors in our pre-screened cohort. And then we also identify new targets for therapy this way for this group of diseases. With that, I would like to end, and I would like to thank the fantastic teams I work with, my research team, and also the diagnostic team in the Center for Medical Genetics at Ghent University Hospital. And then also the clinical collaborators, the many clinical collaborators, which are so important to do the referrals and also to discuss the results of genetic testing. I also would like to thank the funding bodies, and again, also the European Society of Human Genetics, for inviting me. And I'm ready to take questions. Thank you.
Elfride, thank you so much. That's a wonderful presentation, and using so many different approaches to unpick the architecture of these really complex disorders. Got a lot of activity already in the question and answers, so that's great. You've stimulated a lot of thinking from our audience. So if you've got a question for Elfride, do type that into the Q&A box that you'll see at the bottom of your screen. So I'm just going to work our way through some of these questions, if that's okay. So first of all, with regard to the RP-GRIP1 promoter variant, do you which guideline do you use for variant interpretation? Is it the ACMG AMP guideline, or do you use one that's relevant for non-coding variants? What's your thinking around that?
Yes, for the non-coding variants, we use the paper that has been published by Nikki Whiffen and Jamie Ellingford. It's tricky to do variant interpretation and also variant classification for non-coding variants. Here we had a case with the so-called half-solved case where there was a coding variant in the gene. It was a nonsense variant, if I remember well, combined with the promoter variant and also a matching phenotype. So we considered that to be diagnosed patients. So we look at the paper from Nick Whiffen and Jamie Ellingford. ACMG is also working on that. ClinGen is also working on non-coding variations, but it's still more challenging compared to the coding variants.
Yeah, and I'm sure that there are going to be continued developments in that area, trying to work out the best way to interpret those variants. So thank you very much for that question. The next question relates to how you predict the impact of structural variants and asking whether you've used any machine learning based tools to look at those, and if you have, what do you think of those? Do you think that there's any merit in one as opposed to another?
Yeah, so the question is yes. I didn't show data today. We make use of machine learning based tools. There are packages such as the UCIT package, for instance. This is a package that was recently launched and that is oriented toward the interpretation specifically of structural variations and with a particular focus on non-coding structural variations, complex structural variations. So this is very useful to do the interpretation of this kind of structural variants. On top of that, we also make use of Postre. Postre is a tool that has been developed by a Spanish research team, and that is freely available. What is important there for Postre is that there is relevant material, relevant epigenomics data available in the tool. And if you have a disease such as an inherited retinal disease, for instance, it's important that you can link the observation of the structural variant with retinal epigenomic data. We have provided epigenomic data to the team in Spain. So if you work on, say, skeletal disease, then it's important, and you can use the tool if you provide epigenomic skeletal data to the team. So this is another example, and then there are also other AI-based tools, AI-based packages that are also very beneficial and very useful for the interpretation of structural variation. We use a combination of packages. We rarely use one tool. So we, especially with the more complex structural variants, we make use of combination. And then on top of that, if possible, we also look at the epigenomic level experimentally. And this is possible if you can if you have fresh material from a patient, then you can do Hi-C, or you can do Sci-Fi, which is long read sequencing based interaction mapping. This is also a possibility from an experimental point of view, but that is rather in a research context that you can conduct such studies, and that is not very doable in a clinical context.
Very much, Elfride. So we have a question from Seren Alavanda. Thank you for the great talk, and is asking, mentioning Nikki Whiffen, have you identified any cases with an RNU42 variant in your cohort?
Yes, good question. I kept this as an extra slide.
Brilliant.
I had only 40 minutes, so it was a bit too much. And this is also, these are also ongoing findings. So indeed, we did identify, as you probably already saw on bioRxiv or medRxiv, we did identify RNU variants in patients with retinal diseases, more specifically with retinitis pigmentosa. And why did we think of looking at this group of genes? It's a very well-known group of genes. So spliceosomal genes have been implicated in retinal diseases before. Here the brain is not represented here yet on this nice schematic. So we saw all the papers that appeared about the role of the RNA genes in neurodevelopmental disease. And then there was the hypothesis within the European Retinal Disease Consortium if variants in SN RNAs could predispose to retinal diseases. And indeed, they did. So several variants were found in RNA genes, and you see the difference between the variants in blue that have been found in neurodevelopmental disease and then the other ones in retinal disease. And it appears to be, as is the case in neurodevelopmental disease, quite an important contributor to missing heritability in retinal diseases. It's also, so far, we found variants in dominant disease or in monoallelic form in the U4 and U6 SN RNAs. So this is a study that you can read, you can find on medRxiv, and the paper is currently under review in Nature Genetics. So yes, the answer is yes, and this was very surprising and very intriguing. And when I refer to epigenetics and also episignature, for instance, this will be one of the categories of retinal diseases where this will be relevant, as has been done for the neurodevelopmental RNU diseases. I also expect that we will be able to find differences between patients compared to controls, and also the RNA-seq results. There I also expect that this will reveal differences, as have also been observed for the RNU disease, for neurodevelopmental disease.
Absolutely great. I mean, that's fantastic that you had all that prepared, and good luck with the review. There was one other question, but I'm not sure which of your genes it relates to. I think it might have been the RP-GRIP1 case, but the questions from Bernard asking, do you have information on how severe the atrophy was on the OCT of the optic nerves?
For that, yeah, I should go back to the OCT images. It was a non-syndromic form, but I think it was an early onset retinal disease, if I remember well. But I could not answer this specific question about OCT without going back to the
Sure.
OCT images.
Absolutely. Can I just ask, Elfride, how much unknown is there still in your cohort? I mean, obviously, some of the spliceosomal genes will have, increased your diagnostic yield, but is there still plenty to discover?
Yeah, so for our cohort specifically, we did targeted testing of the RNU genes involved, and we did screening of over 350 cases with either dominant or sporadic disease. The yield was rather modest. We have identified mutations in several families and several family members. In total, in our hands, the success rate of when we have done everything, including whole genome sequencing and RNA sequencing and so on, we have an overall success rate of around 60%. So there is still an important gap. There's still an important missing heritability. And I listed some of the potential causes where we have to further look. I expect that in the future, we will have to look more at alleles that are population specific and that are implicated in late onset disease, as we have observed for RPE65. So these novel retinopathies are a bit popping up everywhere. We also see that in other genes. So I expect that for this group of diseases, the late onset diseases that also are sometimes similar to complex diseases, that this will also lead to an increased diagnostic yield. And then also complex alleles that we also now see more and more, and where we also have more insight into the interpretation of these complex alleles using long-read sequencing will also help us to interpret genetic causes of inherited eye diseases. And for this, we have a nice example, for instance, in ocular albinism, oculocutaneous albinism, where we see that it's a very complex combination of alleles, including complex alleles, leads to predisposition of disease. And this is very hard to interpret using short reads technologies. And for this, to solve this problem, to address this, long read sequencing will be very beneficial.
Fantastic. Thank you very much. Elfride, thank you so much for sharing your expertise. A wonderful, clear and interesting talk, which I'm sure our audience has really enjoyed and stimulated a number of questions. And I'm sure that it will help people to go away and think of how to apply these approaches to their own cohorts.